I just finished taking my second to last final of the school year, and you guys are about to go into paper one of the IB Bio test. I hope you guys do well. I thought it would be helpful for me to quickly go through option A in preparation for paper three tomorrow. Okay, so let's get started. If you guys remember, where we first started was talking about neural development. So this is where we're mostly saying how does the brain and spinal cord develop in the womb. There's a couple of structures that you guys want to know and some figures from the textbook that you want to focus on. So mainly what you want to look at is this picture here. This picture is showing the three layers of tissue that form pretty early on in embryonic development. And those layers are the ectoderm, the mesoderm, and the endoderm. So the ectoderm is the green layer, the mesoderm is the middle layer, and the endoderm is the innermost layer. So we're outside here and then moving inside here. What you guys need to know is that the neural tube is one of the first things that's going to develop, and the neural tube is the thing that's going to turn into the brain and the spinal cord. So in order to make the neural tube, we start with this area of the ectoderm called the neural plate. So the neural plate is signified by this black part here. It's the middle of the ectoderm. So it's imagining like it's on the back of the embryo. So the neural plate is going to differentiate these cells so they know they're going to become the neural tube. And then going from neural plate, the neural plate cells are then going to form the neural groove, which basically means they're going to push down. And then the two sides of the neural groove are going to fuse together to make a tube. And that tube is going to elongate along the back side of the embryo's body. So it's able to make the spinal cord and then also the brain. And then when that happens, the ectoderm sides will fuse together, so we'll have a continuous ectoderm and then the neural tube formation there as well. So you guys should generally know those steps, that you have three layers: ectoderm, mesoderm, and endoderm. Ectoderm's on the outside of your body, endoderm's closest to the inside of your body, and the ectoderm is going to give rise to the neural tube. Okay, the other thing you guys want to know is that this picture in purple, this is specifically neural tube formation in a xenopus, which is a like African frog or something like that. Okay, so you guys should be able technically to label the ectoderm, mesoderm, and endoderm of this organism. So like we said, ectoderm is the outside, so this is the green one. Endoderm is the inside, that's the red one, and then mesoderm is in between the two layers. They also want you guys to point out the development of the neural tube. So the neural tube in the last picture was black, but it's actually going to form directly from the ectoderm still. It's just not colored black over in this picture, which makes it confusing. So like we said, the middle part of the ectoderm is going to be the neural plate, and the neural plate cells will then kind of fold, so they end up making the neural groove. And the two sides of the neural groove have to fuse together to make the neural tube, which we see by day 20. And then the neural tube, once it's formed, is going to detach from the rest of the ectoderm layer, and then it's going to elongate along the backside of this embryo to become the spinal cord and the brain. So we can see it pretty clearly on day 22 of frog development. And then on day 36, what we see is that the ectoderm layer has folded up, and it's making a dorsal fin. So dorsal means back, and it's going to make a dorsal fin because the frogs are starting off as like tadpoles, and I guess they have a dorsal fin. The other thing that we want to point out is the developing gut and gut cavity. When you are developing as an embryo, there's kind of two tubes that you have to develop: the neural tube, which is going to form your spinal cord and your brain, and your digestive tube, which goes from your mouth to your anus. And that's going to be developing in the mesoderm. So the mesoderm, which is the red one, that's also going to elongate, and it's going to form the gut cavity. And then the last thing that we want to point out is the notochord. The notochord is going to become the vertebral column. So you've got a spinal cord, and inside the spinal cord, outside of the spinal cord is going to be the vertebrae, which protect your spinal cord. It's a little bit confusing because when I think of neural tube, I think of the spinal vertebrae, and I think of notochord being the spinal cord, but it's the opposite of that. The neural tube is going to be your neurons that are in your spine and in your brain. And the notochord is going to be the bones which make up the spine. And then if you are a chordate, you will always have a notochord. And that's one of the rules that makes you a chordate. But you guys know that because you already studied your phyla for paper 1 today. Okay, so the next thing we want to look at is spina bifida. Spina bifida is that congenital disease that we saw last year where babies can be born sometimes with their spinal cord poking out of their back. And that happens because their vertebrae, which are the bones that enclose the spinal cord, the vertebrae don't close all the way, so the spinal cord kind of slips through that opening of the vertebrae. And they think it happens because the neural tube didn't close all the way and didn't elongate all the way down the back of the embryo when it was forming. So when that happens, the spinal cord is going to poke itself out a little bit. It'll make it harder for the notochord to enclose the entire spinal cord, and the spinal cord will slip out of the back. And that can cause, if it's really serious, like neural defects. But most of the time they're able to fix it because it's usually going to be in your lower back, so the further down part of your spinal cord. So they'll usually fix it by kind of poking that spinal cord back into your body and then maybe putting like a skin graft on top of that. And I think that's all you guys need to know. Alrighty. The other thing we want to talk about really quickly is migration of neurons and development of axons. So once all of the cells in your brain generally decide what they're going to become, if they're going to be spinal cord, if they're going to be part of your cortex, which is the two halves of your brain that you usually think about as being your brain, are they going to become part of your midbrain, like your medulla and your pons? Are they going to become part of your cerebellum, which does your balance? So all your brain cells basically start to move around to where they're going to end up being. So if they're like going to be like optic neurons, they have to move to the back of your head where your occipital lobe is. So what they basically do is have like these little feet around the center of their cell, and they kind of like claw their way or walk their way from like the center of your brain out to their final positions. So like we said, if they're going to deal with visual stimuli, they're going to move to the back of your head where your occipital lobe is. If they're going to deal with like auditory stimuli, they'll move to the sides of your head, like close to your ears where your temporal lobe is. I think that's all we really care about, is that they're going to kind of do contractile actin filaments, which I imagine is like little feet of your cells moving from one location to another, so they can say, this is where I'm going to live, and I'm going to mature here, and then I'm going to get really good at my job. And once they're mature, they're going to develop their axons and their dendrites. So when they're moving, they pretty much just have a cell body. So once they get to their location, they're going to start to develop their axons, and that's going to be in response to chemical stimuli. So they might get some sort of like visual stimulus, which leaves behind like footprints of neurotransmitters, and then the axon is going to grow out that way. Each neuron only has one axon because it would get really confusing for our information processing if we sent information all over the place. But it also might be branched. So it's unidirectional. We can't get information in and out through the axon, just information out. And then we always have lots of dendrites around the cell body that allow it to pick up stimuli from a lot of other neurons around it. And like we said, this is all going to depend on chemical stimuli. So where there are chemical footprints being left behind, or like chemical shadows, which are caused by sensory input, the axons will grow out in that direction. Okay, and then some axons grow beyond the neural tube, reach other parts of the body, and this would be like an example if we're talking about, for example, motor neurons. Motor neurons have their cell body inside the central nervous system, but their axons climb out to the peripheral nervous system. So if you guys recall, there's central and peripheral nervous system. Central is going to be your brain and your spinal cord. Peripheral are all the other nerves. So axons can be super long because they start in your brain and then will climb out like down to your arms and connect to like your biceps and triceps or something like that. But most of those are going to be your muscle neurons. And then some of them will be neurons that connect to your glands, so like connecting to your like adrenal gland or your pancreatic gland to tell that gland to secrete whatever hormone it makes. Okay, and then we've talked about synapses. Synapses are the connections between the presynaptic axon and the postsynaptic dendrites of the next neuron. And we said that every time there's a signal that's sent between an axon of one neuron and the dendrite of the next one, you leave behind like this footprint of neurotransmitters. So neurotransmitters is how, or are how, neurons communicate with each other. So if those pathways are being used, neurotransmitters are going to have to hop across the synapse, tap the next guy, and then go back. But they leave behind kind of a shadow or a footprint, so we can kind of identify, or our bodies can identify, which synapses are the most useful. And those ones are going to be strengthened and they're going to be maintained, and the ones that are not useful are going to kind of prune themselves or be cut away because we're not using them and they're not useful. So we end up just strengthening the ones that are the most useful. So the idea behind that kind of reminds me of like, I don't know if this is true, but if you've been to the University of Maryland, they have this big lawn in front of their library, and supposedly it was just a big lawn of grass. And then they just kind of like let people walk from one side to the other and make their own paths. And we can tell where people have walked because the grass has been eroded there. And then whichever paths were formed because people are walking across this lawn, they put down sidewalks because those were the areas that people used the most. And then anywhere where the grass wasn't eroded, they didn't put a sidewalk down because that's not where people walked to try to get from one side to the other. So that's kind of how I imagine the development of synapses. You have all these synapses, the ones that have the shadow on them is the ones that have been used, and then all the ones that don't, they're not being used, so we get rid of those synapses. And it's just kind of like the idea of like if this road is not being used, there's no sense in using energy to maintain it. We just want to do a really good job of like making sure like 270 and 355 and like East-West Highway are maintained instead of like side roads that nobody uses. That kind of idea. I think that's all I kind of care about talking about for development of synapses. So in this picture, it's just showing here's our cell body of the postsynaptic neuron, and then the green ones are all of the axon terminals that can connect to it and then send messages. And like we said, synapses that are not used are eliminated, and this happens pretty early on. Neural pruning starts to peak at like 8 months, and then it'll continue, and then you kind of end up with all the neurons pruned that you haven't used around the time that you're like 20-ish. So you can use the idea, use it or lose it for that. And like we said, neural pruning, we already covered this. We remove the dendrites and axons that aren't being used through the process of apoptosis. And then the brain is also really plastic pretty early on in your development because things are still trying to figure out which neurons are going to be used, which synapses are used. Your neurons are still deciding exactly what they're going to become. So there's a high plasticity up to the age of 6. So this is when you hear about maybe like kids who have epilepsy. It's a pretty drastic thing to do, but sometimes doctors will do a hemispherectomy where they cut out one of the hemispheres of your brain before your age of 6 or close around that age. Um, and that will sometimes stop the epileptic seizures. Um, and that can be possible because what's left is going to be able to specialize to take care of the functions that the brain side that was cut out was doing. And so around the age of 6, you can still reassign your neurons to do new jobs, and then that plasticity decreases more and more after we go at the age of 6. So you would not suggest somebody who is like 35 years old to get a hemispherectomy because their neurons are already kind of set in their ways and they've already done neural pruning and their brains are less plastic. Not completely plastic, because people who are like 35, even though they can't do the hemispherectomy, they would recover, for example, from like a stroke better than somebody who was 75, because they still have some plasticity where they're able to like retrain their neurons after something like deprivation of oxygen, which occurs during a stroke, would cause. Okay, so plasticity is really important for repairing damage. And we just talked about strokes. So stroke occurs when a clot forms in the blood vessels that are trying to feed your brain. So this might be a blood clot, which is most common, but it also might be like an atherosclerotic clot. If you guys remember atherosclerosis, that's when you form these like lipid plaques on the sides of your blood vessels. If those plaques break away, they can get into smaller and smaller blood vessels and clog those blood vessels. And when that happens, we call it an embolism. And then you can also form a blood clot, which travels up to your brain, and that we would call a thrombosis or a thrombus. The main type of stroke that most people have is called an ischemic stroke, and that means that you are depriving your brain from oxygen. And when your brain cells don't have oxygen for like 2 or 3 minutes, they start to die. You can also have another type of stroke. It's called a hemorrhagic stroke, where the blood vessel breaks, so you start bleeding out, and then your brain is also not getting oxygen for that reason, because the blood's not getting to your brain, it's leaking out of a blood vessel. Your brain uses like 25% of your oxygen and glucose, so it's a pretty small organ. It's only one organ that's using up 1/4 of all that energy and all that oxygen. So when it doesn't get all that energy and oxygen, situation is going to get serious pretty quickly. And like I said, the younger you are, the better you would recover from stroke. Okay. The next thing we want to talk about is brain structure. So going forward, now the brain's all developed and all of that. We talked about the neural tube and all of that. We have like 86 billion neurons. Brain's like the control center, and it receives input from your sensory organs, your eyes, your ears, your nose, your tongue, your skin, etc. And it is connected to your spinal cord. So the brain gets sensory input from all your sensory organs and information up from your spinal cord. And the parts that you guys want to know are in this picture. Obviously, the skull is going to protect the brain. The cerebral hemispheres, or you can call them the cerebrums. You have 2 of them, the left and right one, which is the gray that we see there. That's going to be cut into lobes or divided into lobes, which remember frontal lobe, parietal lobe, temporal lobe, and occipital lobe. And then you've got the pineal gland. The pineal gland is the one that makes melatonin for your sleep-wake cycles. The hypothalamus, which is going to control secretions from your pineal gland, from your pituitary gland, and then from the other glands of your body. So it's a controlling gland. It also does things like your sex drive and your thirst and your hunger regulation. Your cerebellum is going to do like motor and coordination. And your medulla oblongata, if you recall that does a lot of autonomic controls. So like your gut muscles doing peristalsis to do digestion, controlled by your medulla oblongata, your respiratory rate controlled by your medulla oblongata, so you don't have to think about breathing, your medulla oblongata is doing that for you. Your blood vessels dilating and constricting, medulla oblongata is going to do that for you. Your heart muscle pumping, medulla oblongata also does that for you. And it also control your pupil reflexes. So we were saying that if you have like a concussion, one of the things that they do is shine light in your eyes, and if your pupils don't get smaller, then you might have brainstem damage or medulla oblongata damage. And like we said, when you shine light into somebody's eyes, their pupils get smaller because there's so much visual stimuli that's going into their eyes that we make the pupil smaller because we don't need to receive so much light. And then when you're in the dark, your pupils get wider in diameter, so you can try to receive more light because there's so little stimuli. But if your pupils can't change based on how much light is available, it might be because you have medulla oblongata or brainstem damage. Okay. And then we go down into the spinal cord, and like we said, the spinal cord is protected by the vertebrae. The vertebrae come from the notochord, and the spinal cord comes from the neural tube. And then we got pituitary gland. Pituitary gland makes a lot of our hormones. It makes ADH, it makes oxytocin, it'll make FSH and LH. So all really important hormones that we should know. And moving forward. Picture here of a brain lesion. Picture here of an fMRI, which means a functional MRI. So you can look at what's happening in somebody's brain when maybe they're like looking at a picture, or if they're reading a book, or in this case, if they're having pain that has to do with endometriosis. You guys do want to know examples of brain functions. You guys want to know the visual cortex is in the back of your brain in the occipital lobe. You also want to know Broca's area, which is in the frontal lobe, and the nucleus accumbens, which is the area that produces dopamine in response to it's like a reward pathway. Okay. Broca's area produces speech. You guys also might have learned if you took IB Psych or AP Psych, Wernicke's area interprets speech. So Broca's area produces speech. It's like the motor part of speech. And Wernicke's area interprets or receives that speech and figures out what somebody is saying. And I don't know where I was going with that, and that's Wernicke's area. Broca's area is in the frontal, close to the frontal lobe, or within the frontal lobe, and Wernicke's area is more with the temporal and parietal lobes. And then, like we said, nucleus accumbens is going to be the pleasure or reward center of your brain. It's going to produce dopamine when you eat food, so you keep doing that. But that's where a lot of drugs will end up influencing your brain, is in the nucleus accumbens or accumbens. Okay. And then autonomic nervous system. So those are all of the controls that we don't control ourselves. So there's somatic nervous system, which is like our somatic skeletal muscles. And then there's autonomic nervous system. So like smooth muscle and cardiac muscle are controlled by the autonomic nervous system. This is also when we get into parasympathetic and sympathetic nervous system. So hopefully you can picture this. When we do nervous system, we divide it into two parts: central nervous system and peripheral nervous system. Central nervous system is your brain and your spinal cord. Peripheral nervous system can be broken into somatic, which we control, and autonomic, which we don't control. And then autonomic can get divided into parasympathetic and sympathetic. So we have like a tree, which you guys hopefully can imagine. Okay. Parasympathetic is rest and digest, and sympathetic is fight or flight. Like the medulla we already talked about, it does swallowing and breathing and heart rate, all the things that we don't actually have to think about. So there's a center in the medulla that regulates all that stuff. For example, the cardiovascular center in the medulla, blood passes by the medulla and it's able to detect the blood pH, which can tell you how acidic your blood is or how basic it is. And that's going to be a measurement of how much oxygen you have and how much CO2 you have. The more CO2 you have in your blood, the more acidic it is. And the more oxygen you have in your blood, the less acidic it is, because there's got to be a balance between how much oxygen and CO2 there is. And that's going to add up to 100. So if there's lots of CO2, there has to be low oxygen, and if there's lots of oxygen, there has to be low CO2. And our blood pH is about 7.4, and that's what our medulla is going to be able to detect. So if you have acidic blood and it's going past the medulla, that means it has lots of CO2. And what your medulla will cause you to do is increase your respiratory rate, so you breathe off more CO2. So that would be like hyperventilation. And then the opposite would happen if your blood is too basic. And then, if you guys recall, you've also got the SA node, which sends a signal to the AV node, which sends a signal down the bundle of His, which sends a signal to the two sides of the heart for the Purkinje fibers. That's also going to be controlled by the medulla. Okay. And then we already talked about pupil reflex and brain damage before. So if you shine a light in somebody's eyes after they have a concussion and their eyes are not responding by changing shape of the pupils, it's a sign of medulla oblongata brainstem damage. Thank you. Okay. The cortex we just talked about before. We've got the frontal lobe in blue, temporal lobe over your ears in green, parietal lobe kind of like on top of your head, and that in yellow, and then occipital lobe in red, which does visual cortex. Here's our medulla oblongata sticking out, and our cerebellum, which does like balance and motor coordination, that kind of thing. Temporal lobe is going to do auditory integration, so figuring out sounds that you're hearing. So we said like Wernicke's area is here, Broca's area is around here, and the frontal lobe is going to do like judgment, and it's like where your personality is formed and that kind of thing. A lot of times people will talk about from psychology, that guy who, Phineas Gage, that guy who got the like metal pole in his head or something. Parietal lobe is going to do sensory integration, and then occipital lobe is going to do visual integration. And then the frontal lobe and the prefrontal lobe, they are going to be famously underdeveloped in adolescence, which is why your judgment and your insight on life can be questionable, but sometimes it's also better than a lot of adults. Anyway, cortex has gotten bigger and bigger since the first humans, which allows us to kind of take on more and more complex tasks. We also see that it has increased surface area to volume ratio because of the folding, which we call the gyri of the brain. So it just allows us to do more and more complex tasks because we end up having more and more neurons, and they're also getting more and more blood vessels because of that surface area to volume ratio. Okay. And then two graphs, or I guess figures, just comparing brain size relative to body size, and we can kind of see with us, we're pretty far off this line. So even though we're not that big, our brains are pretty big. You guys can kind of expect to see some sort of graph or multiple graphs having to do with option A for paper 3. Okay, we've talked about this before. Left side of the brain gets information from the right side of the body and vice versa. And motor control by cerebral hemispheres. The interneurons of your brain will connect to a motor neuron, which will go out and connect to the muscles of our body. And the left side controls the muscles on the right side, and the right side controls the muscles of the left side. So that's all stuff we already know. And then the green one we don't really have to talk about, but we've seen a homunculus before, which is telling us in the primary somatosensory cortex and the primary motor cortex, these are both parts of the like the parietal and frontal separation. So going back, we have motor right here, and we have sensory here. So it's kind of like a band, like a headband that goes over your head. Motor's in the front, sensory's in the back. And we're just looking at the parts that are dedicated to your different body parts. So for sensing, we can see that like your lips and your face in general take up a lot of space, And then your fingers take up a lot of motor space, which is why our hands are really good at fine motor coordination. And our toes are not as great at fine motor concentration because they get a much smaller chunk of the homunculus. But this shouldn't be super important. The green stuff is not super important. We've already talked about how the brain uses a ton of energy and oxygen and glucose. So I think I said like 25%. Yeah. So us, we're about 20%, even though most vertebrates only use 10%. So our brains are super smart and use up a lot of energy, and small children and infants use up even more energy because they're developing their brains. Okay, and that leads us over to perception of stimuli, which is the last one, maybe second to last one we talked about last year. This is the chapter where we're talking about how we get information from our eyes and how we get information from our ears. So you guys definitely want to know those structures. So our brain is kind of sitting inside of our head, and it doesn't really get any information on its own. So it has to get information from our sensory receptors, which are going to be close to the outside of our body because we need to know what's going on outside of our body. So in your skin, you have mechanoreceptors, which respond to movement and forces. You would also have mechanoreceptors like in your ears to be able to tell you the positioning of your body. You have chemoreceptors, which respond to chemical substances. So you have chemoreceptors like in your nose to be able to tell different smells. Thermoreceptors respond to heat, so those will mostly be in your skin as well. Photoreceptors respond to light, those are in your eyes. You guys want to know these because they might ask you guys to name like 2 types of receptors and what they are receiving as their stimulus. Okay, and then all of these receptors are then going to be linked up with a sensory neuron. So the other thing you guys want to know is a basic reflex loop. And we already talked about olfactory receptors just now, which are chemoreceptors, and we have a bunch of different olfactory receptors, each of which can detect one particular odorant or smell. But then most smells, of course, are made of many odorants, and that's why there's so many unique smells in the world. And we're pretty good at this, but of course, things like dogs, better. Okay. Do I want to look at anything else? No, I don't look at anything else. Okay, what we want to focus on is structure of the eye. But before I do that, I want to really quickly talk about how we said we have a receptor. So imagine a reflex loop over here. We've got a receptor. Receptor goes to a sensory neuron. Sensory neuron goes to an interneuron, which is going to be in your brain or your spinal cord. Interneuron processes the information it just got from the sensory neuron, like, oh, there's a lot of light, or there's a lot of sound, or there's this smell. And then the interneuron communicates to the motor neuron, and the motor neuron is going to communicate to what we call an effector. An effector is a muscle or a gland. So either move this muscle or secrete stuff from this gland. So at the very least, whenever we talk about receiving and responding to information, it's going to take 5 players: the receptor, the sensory neuron, the interneuron, the motor neuron, and the effector. And if it's a really quick response that we don't have to think a lot about, that's going to be a reflex. So like hand over a hot flame. Your thermoreceptors pass that information to your sensory neurons. Sensory neurons pass that information to your interneurons in your spinal cord because reflexes are stored in your spinal cord. And then your interneuron passes information to your to your motor neurons, which then control the muscles in your hand, so you pull your hand away. So that's the most simplistic response that we can have to a stimulus, which we call a reflex arc. But of course, things that are not reflex, that are not like dangerous stimuli that we have like a gut reaction to, it'd be like if you are maybe learning something in class or reading something. If you're reading something, information goes through a photoreceptor, that goes to a sensory neuron, that goes to a bunch of different interneurons in your spinal cord up to your brain, and then probably going to go back to your visual cortex so you can interpret what you're seeing. Then it's going to have to go up to your frontal cortex or frontal lobe so you can actually interpret what you're seeing and make some judgment on it. and then when you're deciding what you want to do with that, then the information is going to go out of your brain, out of the interneurons to a motor neuron, which then connects it to a muscle, which might cause you to like turn a page or something. Anyway, so there's that. That's my spiel on how your brain works. Okay. So looking at the structure of the eye, you guys definitely need to know how to annotate the structure. You shouldn't be expected to draw it. So it says right here, labeling diagram of the structure of the human eye. You guys should be able to go from outside in. So first layer outside is your conjunctiva. That is going to basically like be the part that protects stuff from going into the rest of your body. So the conjunctiva is going to be like the pink part of your eye, and like close to the inner corners of your eye, you have like a little like pink triangle. That's your conjunctiva. Sometimes it can get like irritated and stuff, and it can get like puffy. Going after the conjunctiva, I don't know why I'm saying it like that. I'm pretty sure it's just conjunctiva. Conjunctiva. There we go. Then you go to cornea, and the cornea is going to be the covering of your eye. And then after the cornea, you have this fluid-filled sac called the aqueous humor, and it's going to be where we diffuse oxygen to, because there's not a lot of blood vessels here, and there are cells in your eye that need oxygen to do respiration, to make ATP, and to live. So diffusion is going to occur straight through the cornea into the aqueous humor, so we can carry those nutrients to the other cells of your eye. And then we've got a little opening in your eye called the pupil. The opening is formed by the iris, which is the colored part of your eye. So the iris controls the size of the pupil. So like we said, when you have a lot of light coming in, the iris makes the pupil get smaller, so the opening is smaller. And when you have less light, the iris makes the pupil get larger. Sometimes you guys will see the word aperture, which is like opening. And the pupil is then going to filter all that light into the lens, and the lens is then going to form an image on the back of your retina, or the back of your eye on your retina over here. And that image has to go through the vitreous humor, which is the back set of fluid in the back of your eye, which basically does the same job as the aqueous humor. It just wants to be able to dissolve nutrients and waste to carry them to and away from the cells of your eye. The back layer of your eye has 3 layers to it. It's got the retina, which is the layer that has all the photoreceptors, your rods and your cones. It's got the choroid, which is made out of blood vessels, which then feed your cells so that you can have oxygen and glucose. And then you got the sclera. The sclera is going to form the white of your eye. So it's going to go all the way around your whole eye. And then you've got the fovea. The fovea is where most of the rods and cones in your eye are located, so it's just got the highest capacity right here, but then there's a good capacity all the way around as well. Then you've also got the blind spot, which is where all of the ends of all of the sensory neurons in your eye, they all bundle together and they exit through the back of your eye and form the optic nerve. That area is called the blind spot. So if your lens focused an image on that blind spot, you would not be able to see that object. Okay, so you guys generally want to know these structures, and you want to know like one-ish things that they do. Okay. We already talked about photoreceptors. They're rods and cones. They're located in the retina. Rods do like blacks and whites and grays. Cones do red, blues, and greens. We have 3 different kinds of cones. And then the differences between rods and cones, like we said, rods do dim light, and so they end up doing black, whites, and grays. And cones do bright light, and they do colors red, green, and blue. And the other thing is that rods, they're going to share a bipolar cell, which we'll see in the next page. And cones all have their own bipolar cell, which basically means that rods kind of produce more blurry images, and cones produce better images in our visual cortex. Okay, so what we're seeing here is that our rods kind of function around like 500 as our wavelength, and then we have a cone that works really well at 420. No comments about that, please. That's going to be around our blue light. Then we've got 534, which is going to be the wavelength for our green light, and then 564, which is our color for our like reddish cone, which can go anywhere between like red to like green detection, and then the green does good from like light blue to orange, and the blue cone does well from violet to the other shades of blue. Okay. And then really quickly over here, they're just talking about cataracts, which is when your lens starts to not be clear anymore. So it's really difficult to get light stimuli in. We've talked about red-green color blindness. That's on the X chromosome. It's recessive and it's more common in males, like most sex-linked recessive disorders are. Okay, structure of the retina. You guys do need to know this, and we've talked about this before. You guys should be able to label this and kind of talk about what's happening. So like we said, light comes through your pupil, and then it goes all the way to the back layer of your retina. And then it's going to move forward from there. So here's your red and your blue and your green light coming through. The back layer of your retina is made out of pigmented cells, which can then detect the red, green, and blue. Then the pigmented cells are then going to send their information to a cone cell or to a rod cell. So what we can see here, the cone cells which detect color and the rod cells which do not detect color. We have 3 rod cells that are attached to one bipolar neuron. So this one bipolar neuron receives information from 3 different guys. So it actually forms like blurrier images because it's getting like, it has a lot of work to do on its own. Versus this bipolar cell over here only has like 1 communicator to it. So that's why we tend to see things better in bright light with color rather than dim light with less color because of the bipolar neurons. So that's something that you could put on that works for your mark scheme, but you don't have to. It's kind of a specific thing. So cones, 1 bipolar neuron, rods share a bipolar neuron. But you definitely should know cones do color and bright light reception, and rods do dim and black, white, and grays. Okay, the bipolar neurons then go over to the ganglion cells, and the ganglion cells all connect together to make like one big cord, which is going to form the optic nerve, which we said is the thing that exits through the back of your eye and forms the lines. Optic nerve forms through the back of your eye, and because of that, you have a blind spot. Okay, so you should be able to label this. And like we said, bipolar neurons, they send impulses from the rods and cones, to the ganglion cells. So the bipolar neurons are kind of like the in-between guy between the photoreceptors and the sensory nerves. And this is one of those things where we had to talk about it a couple of times, and I have to refresh myself as well. When rods and cones are not stimulated by light, they depolarize. And when they depolarize, they make an inhibitory neurotransmitter, which does not send signals to the bipolar neuron. And if the bipolar neuron does not get any signal, it becomes hyperpolarized. If you guys remember, that's when it dips below the line. So I'm going to try to pretend to draw a graph here. And if you guys remember, for neurons, they start at -70, so let's say this is -70, then they depolarize because a bunch of sodium goes out, then they repolarize because a bunch of potassium goes out, and then they go below that 70 and they do a hyperpolarization where they can't get stimulated, and then they go back to resting potential, which is -70. So again, it looks like up, down, way too down, and then back to resting. So what we're saying is when cones and rods do not receive light, if they're not getting a stimulus, they polarize. They do this, they go up, depolarize. And when they depolarize, they send an inhibitory neurotransmitter to the bipolar neuron synapse. That causes the bipolar neuron to depolarize, repolarize, and then hyperpolarize. When it hyperpolarizes, it gets stuck in this area where it doesn't send any messages. So then if the bipolar neuron doesn't send any messages, the ganglion cells basically say there's no light. So if these guys don't get any light, they send inhibitory neurotransmitter, causes these guys not to send any signal to the next guys. But if these do get light, they do not send an inhibitory neurotransmitter. So then these guys depolarize instead of hyperpolarizing, and then they do send a message over to the ganglion cells, which then is going to go to your optic visual center of your brain, which is going to be your occipital lobe, which will then be able to process that information. Should we go over that one more time? If you don't need me to, you can just skip ahead by like a minute. Okay, so like we said, light goes in, okay? And if light goes in, the cone cells and the rod cells are not going to depolarize. And if they don't depolarize, they do not send the inhibitory neurotransmitter. And if they don't send the inhibitory neurotransmitter, the bipolar neurons will depolarize. And when they depolarize, they send a message to the ganglion cells. It's like, hey, I'm seeing something. That information goes to the ganglion cells, goes out of your eye through the optic nerve, or by way of the optic nerve, which then goes to the back of your brain to the occipital lobe, and you're able to interpret what you just saw. If there is no light, then these guys, the cone cells and the rod cells, they do depolarize. And when they depolarize, they send an inhibitory neurotransmitter here, which shuts down the bipolar neurons by causing them to hyperpolarize. When they hyperpolarize, they do not send any signals to the ganglion cell. So nothing goes to the occipital lobe, so you interpret it as, I don't see anything. Okay, hopefully that was helpful. Okay, and then we already talked about what the ganglion cells do, and the blind spot, and we already talked about left and right. So here's the optic nerve going out of your left eye, here's the optic nerve going out of your right eye. Having two eyes allows us to compensate for having a blind spot, so that's good. And then they do this thing called optic chiasma, where they do this crossover and go to opposite sides in your occipital lobe. Okay. Now moving on to the ear. You guys again need to be able to label this and know all the names of the structures as well as at least one thing that they do. And I'll make it really easy for you because I'm just going to tell you one thing that they do. Okay, so you've got the pinna. And I'm only telling you one because I only know one. Okay, so you got the pinna. It's going to be the cartilaginous part of your ear, which is going to help funnel sound waves into your ear canal. So we pass through your ear canal, and we've got little hairs here that you guys don't need to know about, but that just helps stuff stay out of your ear canal so we don't get like ear infections. Sound waves funnel into your eardrum or ear canal. They bounce off of your eardrum, cause your eardrum to vibrate. Your eardrum is just like a thin membrane. The vibrations will then cause these three bones, which are called ossicles, which are like mini bones, to vibrate and then pass those vibrations onto each other. So the first bone is called the malleus, the next bone is called the incus, and the last bone is called the stapes. You may also know them as the hammer, the anvil, and the mallet. But IB wants you guys to call it malleus, incus, stapes, so MIS. Then the stapes is going to pass those vibrations into the oval window, and then when stuff goes into the oval window, it goes into the cochlea, and then gets processed inside the cochlea, and the sound waves exit through the round window and then down this tube. I think it's called a eustachian tube. It's the one that gets weird when you like are on a plane or something like that. And then whatever the cochlea is interpreting goes out of the cochlea through the auditory nerve over to your temporal lobe for processing. And then we'll go into it more, but remember the cochlea has these little hair cells in it, and the hair cells have different lengths, and they can vibrate and then interpret different tones. And then they'll pass that information on to the sensory neurons in the cochlea, which then form the auditory nerve. And then you've also got the semicircular canals, also have little hair cells in them. And remember, there's an X and a Y and a Z axis, and they're going to help to tell you the positioning of your head because there's fluid and these little hair cells in the semicircular canal. So when you turn your head, the fluid pushes over those hair cells and it can tell you the positioning of your head. So sometimes when people have like poor balance or like they're dizzy, it might be like a semicircular canal issue. And that's all we need to know. The bones also help with conduction of sound, but I don't think you guys really have to worry about that. They really care about more of this stuff. We would call this the outer ear, the middle ear, and then the inner ear for inquiring minds. Okay, hang in there. We're almost done. Okay, middle ear is going to transmit and amplify sound. Those are going to be those tiny little bones. And then the inner ear does the detection of sound. So outer ear collects sound, middle ear transmits and amplifies sound, and cochlea is going to detect and then also transmit sounds. Like we said, we've got these cells with hairs in them, and the hairs are different lengths, so each length can detect a different type of like tone or frequency, well, tone and frequency, and then they'll activate different hair cells because of their vibrations. Other things I want to talk about, auditory nerve is going to transmit over to the temporal lobe. And then if you have issues with your cochlea, which is supposed to send information to your auditory nerve, which sends information to your temporal lobe, which is then going to get processed by you. Sorry, just making sure this was still recording. That would be really bad if it wasn't. You can get a cochlear implant. And I honestly do not remember a lot about this, but I did recently watch A Silent Place, and the girl had a cochlear implant. Okay, so if the hair cells in your cochlea don't work, hearing aids don't help because hearing aids just increase the sound. So they basically do like the job of the middle ear to try to amplify the sound. But if it's not an amplification issue, it's more of a detection issue, then you would want a cochlear implant. If the issue is the auditory nerve not being able to pass down, not the auditory nerve. If you have a cochlear implant, your auditory nerve works fine. It's more of the hair cells inside of your cochlea don't work fine. If you have issues with your auditory nerve, doing a cochlear implant won't help because cochlea will work and then they can't pass it on to the auditory nerve, which passes it on to your brain. So it's got internal and external parts. It'll have like a little thing on the outside of someone's skull, and then I would imagine that they would insert something into the cochlea through surgery. But the external parts have a microphone, also to amplify sound, and the microphone is going to go and be connected to the transmitter. The transmitter is going to process sound, which go into the receiver and the stimulator, which will then stimulate the inner cochlea parts. They've got a receiver that pick up signals and the stimulator, which converts the signals into electrical impulses. Those electrical impulses are going to be sent out the auditory nerve. So microphone and transmitter are outside. Microphone picks up sound. Transmitter sends the sound over to the cochlea. So this part is outside of your head. This part is on the inside of your head. And then receiver is going to get information from the transmitter, and the stimulator is going to convert the sound into electrical signals, and the electrical signals will be passed on to the auditory nerve, which will then go to your temporal lobe for interpretation. Okay, I just said a lot of things. I don't know if I said them clearly. So again, microphone, transmitter, external. Microphone picks up sounds, passes it to transmitter. Transmitter passes that over to the receiver and the stimulator, both of which are inside of your head. The receiver gets information from the transmitter and then passes it over to the stimulator. The stimulator converts the signals into electrical impulses, which can be carried by the auditory nerve and interpreted by your temporal lobe. Okay. There's that. Let me scroll down. You guys don't really need to worry about this green box. And like we said, head movements in the semicircular canals, there's 3 of them, and they work in the X, Y, and Z axes. And like we said, they're filled with little cells that have little hairs on them, and they've got like a blob of fluid on top of that, which we call a cupula. And then when your head moves, it causes the fluid of the cupula to move. Um, and this can be detected by the hair cells, which send impulses to your brain for which direction you're in. I have a better picture than this that is in my slide. So maybe you still have these. We've talked about this before. You've got the hair cells, you've got the little hairs coming out of them, you've got a cupula, which is like a gel on top, and then you've also got fluid outside of that. So when you turn your head, the fluid flows over the cupula. The cupula then bends the little hairs inside of your cupula, and then all of your hair cells are going to be attached to a sensory nerve. So, like we said, receptor, here's the nerve, and then this all sends information to your brain to tell you the direction of your head. And I think that's all I wanted to talk to you guys about. Okay, so that's A1, A2, and A3. And I'm going to pause there because this was kind of long, and then I'll do another one for A5, and then A4 and A6.